Assalamu alaikum. So we are continuing our topic, neoplasia, and today our topic is the molecular basis of the cancer. So there are the 4 classes of regulatory genes are the principal targets of genetic damage. What are the proto-oncogenes? Genes that code for the proteins involved in the control of cell growth, like growth factors, growth factor receptors, and signal transducer. Mutant alleles dominate, which transform the cells despite presence of normal counterpart, is equal to phenotype affected even if one allele is present. The tumor suppressor genes, second one, the genes that produce products that inhibit cell growth and control the G1 to S1 phase of the cell cycle and nuclear transcription. The both normal alleles must be damaged for transformation to occur and results in the recessive oncogenes, which leads to the malignant phenotype, which only develops if both alleles fail to suppress their growth. Third one is the apoptosis genes that regulate the programmed cell death. Examples are the PEX apoprotein, apoptosis gene. These are activated by the TP53 if DNA damage is excessive. The BEX protein inactivates the BCL2 anti-apoptosis genes, and the mutation of TP53 leads to the inactivation of the BEX, and there is no apoptosis. The fourth one is the genes involved in the DNA repair. The loss of activity leads to the DNA instability, which ultimately results in the somatic mutation in oncogenes or tumor suppressor genes, and both alleles must be inactivated. Carcinogenesis is a multi-step process at both the phenotypic and the genetic level. The phenotypic attributes of the malignant neoplasm are required in a stepwise fashion called as the tumor progression, and the progression results from accumulation of genetic lesions, means multiple mutations. Look at this picture. This is the normal cell. When first mutation occurs, the cell seems normal but is predisposed to proliferate excessively. And when second mutation occurs, the cell begins to proliferate too much, but it otherwise looks normal. This is normal, the second mutation. When there is a third mutation, the cell proliferates more rapidly. It also undergoes structural changes. You see the structural changes of the cells. And when the fourth or the later mutations occur, the cell ultimately changes into malignant cell. Cell grows uncontrollably and looks obviously deranged.
Can you hear me? Can you hear me?
So, as we discussed earlier, the fundamental changes in cell physiology that determine the malignant phenotype are the first one, the cell sufficiency in growth signals, insensitivity to growth inhibiting signals, evasion of apoptosis, limitless replicative potential, sustained angiogenesis, ability to invade and metastasis, and defects in DNA repair. So first of all, we discuss self-sufficiency in growth signals. What is oncogene? The genes that promote autonomous growth in cancer cells, which is and their unmuted counterpart is called as the proto-oncogenes. These are created by the mutations in proto-oncogenes. Oncogenes are created by the mutations in the proto-oncogenes. The product similar to normal counterpart but lacks important internal regulatory elements which indo means empower or enable the cell with self-sufficiency in growth. So self-sufficiency in growth signals. Normal cell proliferation, the binding of growth factors to the receptor. There is the activation of singular transitional protein. Transition protein on inner leaflets of plasma membrane, which leads to the transmission of signal across cytosol to the nucleus, which ultimately causes the induction and activation of nuclear regulatory factors, which initiates the DNA transcription, entry, and progression into the cell cycle. The activation of the oncogenes in human tumor. How it possible? How it occurs? By three ways: the point mutation and the deletion. Point mutation and the deletion. Example is the RAS protein, which occurs in the CA bladder and cholangiocarcinoma. The second one is the chromosomal translocation. The example of the chromosomal translocation is the 95% of chronic myeloid leukemia, where the CABL proto-oncogene on chromosome 9 is translocated to the chromosome 22. The other example is this 75% of Burkitt lymphoma translocation of CMYC proto-oncogenes from chromosome 8 to chromosome 14. The third method of activation of oncogenes in human tumor is the gene amplification. What is gene amplification? It is the chromosomal alteration that results in increase in the number of copies of gene found. Example of this gene amplification is the neuroblastoma having NMYC HSR region. The role of oncoproteins. So, these are the steps in signal transduction for cell proliferation by oncogenes. First of all is the growth factor. These are the most soluble growth factors made by one cell type and act on a neighboring cell to stimulate proliferation, called as the paracrine action. Means normally act on other cell other than the one who synthesizes. यानी यहाँ पे वो synthesize होता है, वो वहाँ पे action नहीं देता है, वो किसी और cell पे action देता है, and to stimulate its proliferation. उसके बाद तक क्या? The most cancer cells able to synthesize the same growth factor to which they are responsive in an autocrine loop. लेकिन जो mutated और cancer cell होते हैं, they can synthesize growth factor which may respond to it. In this way, the cancer cell acquire growth self-sufficiency. Examples of such growth factors are the platelet-derived growth factor and transforming growth factor. Now, the second step in the signal transduction is the growth factor receptor. The normal transmembrane receptor cytoplasmic tyrosine kinase transiently activated, followed by receptor dimerization and tyrosine phosphorylation. इसमें क्या है कि many oncogenes encoding growth factor receptor which act more commonly by overexpressing of normal growth factor than by mutation. यानी इसमें म्यूटेशन नहीं होगी लेकिन ओवर एक्सप्रेशन ऑफ द ग्रोथ फैक्टर्स होंगे. उसकी एग्जांपल क्या है? ERB1NN एंडोथेलियल ग्रोथ फैक्टर, उसमें क्या होता है? ओवर एक्सप्रेशन इन स्क्वैमस सेल कार्सिनोमा. Oncogenic version which constitute dimerization and activation without binding to the growth factor, which leads to the continuous mitogenic signal to the cell even in the absence of growth factors in the environment. Growth factor receptor, the mechanism of constitutive activation of growth factor receptor. First of all, there is a mutation and gene rearrangement, RET protein. This is normally expressed in neuroendocrine cells. RET means these are the rearranged during transfection. It is localized on chromosome 10. These are the proteins which are rearranged during transfection. These are normally expressed in neuroendocrine cells. There is a point mutation in extracellular domain, the MEN2A. What does mean by MEN? MEN is the multiple endocrine neoplasia. Multiple endocrine neoplasia type 2A, which is seen in thyroid, adrenal, and parathyroid tumor. And other point mutation in cytomlasmic domain, which occurs in MEN2B. Multiple endocrine neoplasia 2B type seen in thyroid and adrenal tumors. And these are most commonly seen in autosomal dominant syndromes. The growth factor receptor, the mechanism of constitutive activation of growth factor receptors. There is a mutation and gene rearrangement, as we discussed earlier. The point mutation seen in the myeloid leukemias. Growth factor receptor mechanism, constitutive activation of growth factor receptors. There is the overexpression of normal forms of growth factor receptors. These are more common: ERBB1, endothelial growth factor receptor gene, seen in 80% of squamous cell carcinoma of lungs, and FRBB2, HER2, NEU, seen in breast carcinoma and adenocarcinoma of the ovary. The third step in the self-sufficiency in growth signal is the signal transducing protein. These, located on the inner leaflet of the plasma membrane, receive signals from outside the cells and then transmit to the cell's nucleus. Most well-studied is the RAS family of GTP binding protein or the G proteins. The RAS oncogene. These are the point mutation of the proto-oncogenes, that single most common abnormality in human tumor, located at cytoplasmic aspect of the plasma membrane as well as the membranes of the ER and Golgi apparatus. The RAS proteins bind guanosine nucleotides. So this is the RAS oncogene. The stimulation of cells by growth factor, which inactivates the RAS, and there is the exchange of guanosine diphosphate for guanosine triphosphate, which activates the RAS protein and stimulate the mitogenic activated protein kinase cascade, and which results in the fluid nucleus with signals for cell proliferation. How it works? RAS is the most common oncogene in the human tumor. What does it mean? RAS means rate sarcoma gene. Rate sarcoma gene seen in most commonly seen in the CA colon, lung, and the pancreas, having two forms, active and inactive. The active form of the RAS protein, which is the GTP, it is the guanosine diphosphate, which is the activated form of RAS protein is the GTP. Sorry, the GTP is the inactive form, while the GTP is the active form of the RAS protein. Both are same to the G protein and take part in signal transduction in on and off switch manner. Normally what happens, the RAS protein active one is inactivated by the GTPase. Which one is the active one? The RAS active one, which is the GTP, which is inactivated by the guanosine triphosphate enzyme activity, while mutated RAS remains unaffected by the GTPase. The mutated RAS factor, which is unaffected by the GTPase, and which results into continuous signal to the cell to proliferation, and common mode of activation in this process is the point mutation. So what happens? The activation of oncogenic RAS leads to the upregulation of which block the cyclin D CDK4 mediated hyperphosphorylation of RB pro cellular sequences. And the RAS signaling might also feed into the ARFP53 pathway to promote apoptosis as well as the reinforced cellular sequences. The mutated RAS trapped in its activated GTP-bound form due to the inactivation of GTP hydrolysis, which results in cell forced into a continuous proliferative stage. इसमें क्या होगा कि the mutated जो होगा, normally क्या होता है कि RAS protein active one is inactivated by the GTPase, while the mutated RAS remains unaffected by the GTPase, and which enfolds the cell into continuous proliferative state. And what are the examples of mutation of KRAS? Is the carcinoma of colon and pancreas, as we said earlier. The mutation of HRAS seen in bladder tumors, and the mutation of NRAS is seen in hematopoietic tumors. The transcription factors. What are the transcription factors? It is transduction signals, the stimulation of nuclear transcription factors, which leads to the binding to the DNA or dimerized for DNA binding, and which leads to the DNA transcription. Transcription factors. The ultimate sequence of signaling through oncogene is inappropriate and continuous stimulation of nuclear transcription factors. There is a growth autonomy occurs as a consequence of mutation affecting genes that regulate transcription. For example, MIC. So there is a growth autonomous. So when there is a cell mutation, cell becomes mutated. It will lead to the uncontrolled growth of the cell. MIC oncogene. The cell signal transduction pathway. What is it? It started from with the growth factors to the nucleus, where it regulates the DNA transcription. The MYC oncogene is the most common of the nuclear regulatory transcription protein found in the human tumors. The proto-oncogene expressed in all eukaryotic cells. Immediate response genes rapidly induced when quiescent cells receive signals to divide. The target gene of oncogenes include ornithine decarboxylase and cyclin D2, which is associated with the cell proliferation. The range of activities modulated by the MYC is histone acetylation, reduced cell adhesion, increased cell motility, increased telomerase activity, increased protein synthesis, and the decreased proteinase activity. The range of activities modulated by the MYC. The selection of origin of replication. There is overexpression which leads to more origin of the replication needed for the normal cell replication. Bypass checkpoints involved in the replication. Reprogramming of the cells into the pluripotent stem cells enhances the self-renewal, block differentiation, or both. What happens? The MYC protein binds to the DNA, which regulates the cell cycle by transcriptional activity, which results in the decreased level immediately after cell enters cell cycle. क्या होता है कि MYC प्रोटीन जो होता है पहले सबसे पहले वो बाइंड होता है DNA से जो सेल साइकिल को रेगुलेट करता है थ्रू द ट्रांसक्रिप्शनल एक्टिविटी और then it will be ultimately results in the decreased level immediately after cell enters the cell cycle. But on the other hand, the MYC oncogene associated with the overexpression of the MYC oncoproteins. There is an autonomous cell proliferation. The persistent expression commonly found in tumors, the translocation, example, the Burkitt lymphoma, amplification, the carcinoma of the breast, colon, lungs, and other carcinomas. So in the Burkitt lymphomas, there is a mutation due to the translocation of chromosome 8 and 14. While in lung, breast, and colon, MYC gene mutated due to the amplification. Okay. So here there is a increased proliferation. The MYC induces cell proliferation, cell transformation, and growth arrest. Then it will lead to the apoptosis. The biological response to CMYC is dependent on the cell lineage and environment. MSC is an intracellular sensor and transducer of the extracellular stimuli. So when oncogenic mutation occurs, the growth factor, cytokine, cell adhesion, contact inhibitions. So these occurs cell growth and proliferation, differentiation block, immortalization, cellular transformation, angiogenesis, migration, metastasis, cell fate transition, apoptosis, DNA damage responses, and genomic instability. Next step is the dysregulated activity of the cyclin and the CTK. First of all, what happens in the normal cell cycle? The orderly progression of cells through cell cycle orchestrated by CDKs bound to the cyclin. The CDK cyclin complexes phosphorylate crucial target protein. They drive the cell through the cell cycle. क्या होता है कि normally cell cycle is under regulates the control of the cyclin and cyclin dependent kinase. यानी normal cell cycle जो होती है under control होती है cyclins and cyclin dependent kinase, which are the A, B, and D. Cyclin D, E, A, and B appear subsequently during the cell cycle and bind to one or more CDKs. In normal cell cycles, there is inhibitors of CDKs. These are SIP or wave family. This inhibits the CDKs broadly. Do you know why do we call it the cyclin? Cyclin, named after because they cyclically synthesize during different phases of cell cycle, and their degradation is also cyclic. Cyclin work together as well as activate together with CDKs. Although all the steps of the cell cycle is under regulatory control, but there is a checkpoint frame from G1 to S checkpoint. There is a checkpoint in the cell cycle. It checks for the DNA damage, which prevents the replication of the cells with defects in DNA. And the second checkpoint is the G2 to M checkpoint, which monitors the completion of DNA and check whether the cell can safely initiate mitosis, and important in cell exposed to ionizing radiation. Cell cycle checkpoints require senses of DNA damage, the proteins of RAD family and ataxia telangiectasia mutated, and the signal transducer CHK kinase family. Cell cycle checkpoints require effector molecules, G1 से लेके S phase तक, P53 and P21, and G2 से लेके M phase तक P53 dependent and P53 independent mechanism. The mutation this regulates activity of the cyclin and CDKs favor cell proliferation. जब mutation होती है in cyclin, particularly in the CDK, in cyclin D, the mutation affecting expression of the cyclin D or CDK4 common in neoplastic transformation are most growth promoting signals in cancers. Following tumors have such oncogenes. यानी जब mutations होती है तो वो most commonly होती है cyclin D में और CDK4 में. CDK frequently mutated or otherwise silenced in many human malignancies, somatically acquired deletion or inactivation of P16, the pancreatic carcinoma, glioblastomas, esophageal cancer, ALL, bladder cancers, and germline mutation of the P16 found in the melanoma. This is the defects in cell cycle checkpoints components are a major cause of genetic instability in cancer cell. अगर checkpoints में अब mutation हो जाती है तो ये सबसे major cause होती है genetic instability की तरफ. Now we move on to the tumor suppressor gene. These apply brakes to the cell proliferation. So they prevent uncontrolled growth. They recognize genotoxic stress and shut down the cell proliferation. Their expression of oncogene in a normal cell lead to the quiescence or permanent cell cycle arrest. For example, is the RB or the retinoblastoma. RB protein is a ubiquitously expressed nuclear phosphoprotein, exists in active phosphorylated state in quiescent cells and an inactive phosphorylated state in the G1 to S cell cycle transition. So in simple words, we can say the mutation of the normal growth suppressor anti-oncogene or tumor suppressor gene results in the removal of the brakes for the growth. Thus, the inhibitory effect of the cell growth is removed and the abnormal growth continues unchecked. The tumor suppressor gene का main function क्या होता है कि brakes provide करता है. If it doesn't work or mutated, then or breaks, there is a removal of brakes for growth proliferation. The inhibitory effect of the cell growth is removed and the abnormal growth or proliferation occurs uncheckedly. So the point of action by anti-oncogene is also G1 to S phase transition. The major anti-oncogenes is we discussing is the RB or retinoblastoma gene, which is located on the long arm of the chromosome 13. It is important in its enforcement of G1. The cells can access the cell cycle either temporarily or permanently, which induces the synthesis. RBC also controls stability of the cell cycle inhibitor P27. The RB gene exists in the active and inactive form. The active RB genes inhibit cell cycle at the G1 state from G1 to S phase. So there is a hypophosphorylated active RB binds to and inhibits the E2F. There is no cyclin E transcription, leads to the progression to H phase inhibited. There is a hyperphosphorylated RB, which is an inactive form of RB, leads to the release of E2F, and there is a transcription of cyclin E with DNA replication and progression through the cell cycle. The absent RB due to the gene mutation. No regulation of E2F transcription factors and DNA replication and continuous progression through cell cycle. Mutation in other genes that control RB phosphorylation can mimic the effect of RB loss. There is the mutational activation of cyclin D or CDK4 facilitate the RB phosphorylation, and the mutational inactivation of CDKIS, which is unregulated activation of the cyclin and CDKs. The loss of normal cell cycle control is central to the malignant transformation. At least one of the following regulators of the cell cycle is dysregulated in majority of the human cancer, which may be P16, INK4A, CDK4, cyclin D, or can be RB. Now, the next gene is the TP53. TP53 gene located at chromosome 17, P13.1, most common target for genetic alteration in human tumors, senses cellular stress such as DNA damage, shortened telomerase, and hypoxia, function as a critical gatekeeper against the formation of cancer, molecular postman, or a guardian of the genome. The P63 and P73 are the collaborators of the P53. The P53 ubiquitously express while P63 and 73 with more tissue specificity. The P63 is essential for differentiation or stratified squamous epithelia, while the P73 with strong proto-apoptotic effects after DNA damage from chemotherapeutic agents. This prevents the malignant transformation by activation of temporary cell cycle arrays, quiescence, the induction of permanent cell cycle arrays, synthesis, and the triggering of programmed cell death, means apoptosis. So, what happens when there is a DNA damage agents or like hypoxia? There is an activation of the normal P53. There is the upregulation of KAT45 genes, leads to the induction of DNA repair. There is a successful repair and cell cycle progress, which happens in daily basis in normal cell. While in mutated cell, the upregulation of CDK1, P21, which cell cycle arrays in the G1 phase. And there is an unsuccessful repair which activates the MIR34 and stimulate bags. And when there is stimulation of the bags, which leads to the apoptosis, and when there is activation of MIR34, either it inhibits the growth promoting genes MYC or CDK4, or inhibit anti-apoptosis gene like BCL2. And there is a question sequence. What happens? As another diagram, DNA damage, the P53 cell cycle arrest, and the DNA repairs occur, and the cell proliferate again normally. But if there is a irreversible DNA damage, there is an increased bags, decreased BCL2, decreased FAS, increased FAS, and other mechanism leads to the apoptosis. These are the summary of mechanism of action of tumor suppressor protein genes. So there is another example, DNA damage agent or hypoxia leads to the cells with mutation or loss of P53. DNA damage leads to the no activation of P53 dependent genes, which ultimately results in cell cycle progression, no DNA repair, no synthesis, and results in mutant cell expansion and additional mutation, and ultimately there is the formation of malignant tumor.
Since it is not necessary to know the microbiome, but it is for your knowledge to know the cause of disease. When you know the cause of the disease, it is
Homozygous loss of the P53 occurs in virtually every type of cancer. In most cases, the inactivated mutation affect both P53 alleles and are acquired in somatic cells. Approximately 80% of P53 point mutation present in human cancers are located in the DNA binding domain of the protein. In the majority of the tumors without P53 mutation, the function of the P53 pathway is blocked by the mutation in another gene that regulates the P53 function. So MDM2 and MDMX stimulate the degradation of the P53, and there is overexpression in malignancy. The amplification of MDM2 leads to the human sarcomas. MIR34 microRNA is important to P53 response, results in the target including proliferative genes such as cyclins and anti-apoptotic genes. There is the inhibition or blockage of MIR34, which leads to the impaired P53 response, ectopic expression of MIR34 without P53 activation, and results in the growth arrest and apoptosis. So what happens?
now the last topic is the topic why? Because endometrial hyperplasia, if it is not treated, not can lead to endometrial hyperplasia.
आवाज बनियन होला है ना जी आवाज अला बनियन होला है ना
Associated with prolonged
Hello?
hyperplasia results from prolonged unopposed with such conditions include
हेलो बेटा आवाज आ रही है ना हेलो बेटा आवाज आ रही है आ रही बेटा
नो माइक बंद
ठीक है अच्छा तो आज का टॉपिक हम लोग यहीं पे क्लोज करते हैं नेक्स्ट टाइम हम लोग देखेंगे एक और जीन यानी यहाँ तक आज हमने किया ट्यूमर सप्रेसर जीन। एनी क्वेश्चन या इसे कुछ आसान करने के लिए मुझे और बनाना पड़ेंगे? अच्छा हम लिखी मोकले ये गद्दी तो गलन समझ में न पहुंचे। Yes, beta, it is a quite lengthy chapter. लेकिन what to do? We have to. What we supposed to do? Questioning. You can ask question in the meanwhile in the chat. जी बेटा कोई क्वेश्चन है आपको? हेलो बेटा आवाज आ रही है? जी बेटा आर यू गेटिंग मी? एनी क्वेश्चंस? तो इससे मैं क्या समझूं कि कोई क्वेश्चन नहीं है? बेटा आप लोग अपने क्वेश्चंस टाइप भी कर सकते हैं और पूछ भी सकते हैं। आपको अनम्यूट किया गया है। Okay, so we are ending the session here for today. Next time, most probably, the second last lecture of this chapter. Okay, so goodbye for now. Thank you.